Good afternoon to all. In the previous class, we are discussing the process of megasporogenesis, or how the female gamete is developing, and we have already seen that female gametes are formed inside the ovary or gynecium, and inside the gynecium the nucellus develop, and finally it develops into an ovule with two integuments. We complete the topic up to that. Then you have to write the notes. Class result ചെയ്യുന്നതിന് മുമ്പ് you have to write the notes. എന്നാൽ മാത്രമേ ക്ലാസ് ശ്രദ്ധിക്കാൻ പറ്റൂ. And the development of ovule we have seen in the last class. Each ovule have a mass of tissue called a nucellus, and the nucellus is covered by one or two layered covering, and this covering is called integument, and the tissue is called parenchymatous tissue is called nucellus, and the stalk of nucellus is called funiculus, and the point where the funiculus joined with the ovule is called chalaza, and the upper end of the ovule or the other end of the ovule where there is a small opening, that small opening is called micropyle, and this end of the ovule is called micropylar end. This opposite end of the ovule is called chalazal end, where the chalaza is present. So this end is called chalazal end, this end is called micropylar end. So this is the structure of ovule, and this mass of tissue, you remember that this is a mass of parenchymatous tissue, and this mass of parenchymatous tissue is called nucellus. Chromosome number, or it is usually diploid in number. So this is the ovule with the LS of the ovule. I just represent one integument only. One or two integuments are present. If one integument is present, it is called inner, and some may be outer one may be also present. So this is the structure of ovule. See, up to this, female gametes are not developed. So we can see in this class how the female gamete or egg or embryo sac develop. Female gametes are developing inside the nucellus. You remember that this nucellus is a mass of parenchyma tissue like that. Yes, we are doing like that only. Just a mass of parenchyma tissue is called nucellus. And see how the female gametes are developing inside the nucellus. Today we can see how the female gamete or megasporogenesis takes place inside the ovule. So remember that female gametes or megaspore, female gamete is developing inside the embryo sac or inside the nucellus. See, this can be represented sequentially by diagram. You can copy the diagram, then after this ovule represent inside the nucellus, represent like that. It consists of a mass of tissue, and towards the micropyle during the megasporogenesis, during the development of female gamete, in the nucellus, remember that this is a mass of tissue. In the nucellus, one cell towards the micropyle differentiate. It becomes more clear with the thick cell wall, full cytoplasm, and clear nucleus. One cell towards the micropyle differentiate, or it becomes clear with the full cytoplasm, thick cell wall, and clear nucleus. One cell differentiate, become more clear. That one cell is called as megaspore mother cell. That one cell is called a megaspore mother cell. It is diploid in nature. One cell differentiate. That one cell is called a megaspore mother cell. Then that megaspore mother cell divide by meiosis. It divide by meiosis. And that represent once again this megaspore mother cell divide by meiosis into 4 megaspores. Can represent that. It divide by meiosis into 4 megaspores. See, by meiosis, one cell divides into 4 cells. So the megaspore mother cell which is diploid divides into 4 megaspores. These cells are called megaspores, which are haploid in nature. So megaspore mother cell divided by meiosis into 4 haploid megaspores. Of the 4 haploid megaspore, 3 destroyed. See, of the 4, 3 destroys. Just represent like that. 3 destroyed. See, 3 towards the anyhow 3 cells destroyed. 3 cells destroyed. 1 survived. This 1 cell survived. 1 megaspore survived. The 4, 3 destroyed, only 1 survived. This is a surviving megaspore. 1 remains. And this 1 surviving megaspore enlarged into embryo sac. This surviving megaspore developed into embryo sac. This type of embryo sac development is most commonly seen in angiosperms. And this kind of embryo sac development is called monosporic type of embryo sac development. These questions are usually asking for competitive examination. Monosporic, bisporic, tetrasporic, etc. Here in most of the angiosperms, 3 destroyed. Remember that 3 destroyed, 1 survived. This 1 surviving megaspore developed into embryo sac later on. And this type of embryo sac development is called monosporic type of embryo sac development. So 1 survive. Then the nucleus of the megaspore, or this megaspore enlarged in size. This megaspore enlarged in size. And its nucleus, so that is a haploid nucleus, divide 3 times by mitosis. Divide 3 times by mitosis. See, I will represent that. Nucleus divide 3 times. 1. First into 2. Next to mitosis, 2 into 4. Again another mitosis. Again another mitosis. 4 becomes 8. See, 3 mitosis. 1 into 2. 2 into 4 and 4 into 8. So nucleus divide 3 times by mitosis into 8 haploid nuclei. Remember that meiosis is over, then all are haploid. Then 8 haploid nuclei. Of the 8 haploid nuclei, nuclear arrangement takes place. So listen here carefully. This is the nucellus. This embryo sac enlarges in size. See, embryo sac enlarges in size. Again enlarges. 8 nuclei are there. Of the 8 nuclei, 3 move towards the micropyle. 3 move towards the micropyle. 3 move towards the chalaza, opposite end. 2 remains at the center. So listen here again. 8 nuclei are there. They are remaining in common cytoplasm. And of the 8 nuclei which remains in common cytoplasm, 3 move towards the micropyle, 3 move towards the chalaza, and 2 remains at the center itself. Then cell wall formation takes place around each nucleus. See, the cells are covered by cell wall. Cell wall formation takes place around each nucleus. So 1 cell, 2 cell, 3 cell. Here also cell wall formation takes place around each cell except at the center. Around the center, cell wall formation does not occur. So of the 8 nuclei, 3 remains at the micropylar end. 3 move towards the chalazal end. This point is called chalazal end. 2 remains at the center. And of the 3 at the micropylar end, the central cell is called egg cell or female gamete. The cells on either side are called as synergid cell. Synergids. So central egg cell and two synergids. Three at the opposite side are called as antipodal cells. Antipodal cells. And two remains at the center. They are together called polar nuclei. Of the 8 nuclei, 8 nuclei, 3 arranged at the micropylar end, and of that central cell is called egg cell or female gamete. Side cells on either side are called synergid. So one egg cell and two synergids are together called egg apparatus. Egg apparatus. So two synergids and one egg cell and two synergids are together called as egg apparatus, and 3 cells on the opposite side are called antipodal cells, and 2 remains at the center. They are called as polar nuclei, which later join to form secondary nucleus. So this is the mature embryo sac. So this embryo sac at this stage is called female gametophyte. Embryo sac at this stage is called female gametophyte. You can call it as female gametophyte, embryo sac or female gametophyte, because gametophyte, you know, gamete containing structure is called gametophyte. So female gamete is present inside the embryo sac, and this structure is called female gametophyte. Fully developed embryo sac is called female gametophyte. So this female gametophyte or embryo sac is a 7-celled 8 nucleated structure. 7 cell, 1, 2, 3 cells, 1, 2, 3 cells, and this can be considered as a central cell. So 6 cell, 7 cell, 8 nucleated, because the central cell is with the 2 nuclei, binucleated structure, 2 polar nuclei. So this is the female gamete. 2 synergids, 3 antipodals and 2 polar nuclei. This is the structure of fully developed embryo sac or female gametophyte. I think I will revise once again how the development of female gamete takes place. First of all, inside the nucellus, a cell differentiates. That cell is called a megaspore, mother cell. This megaspore mother cell is diploid. This divided by meiosis into 4 megaspores, which are linearly arranged. Of the 4 megaspores, 3 destroy, 1 survive. Surviving megaspore enlarges into embryo sac, and embryo sac nucleus, megaspore, nucleus of the surviving megaspore, divided by mitosis 3 times. Only nuclear division takes place, karyokinesis takes place, cytokinesis will not take place. So nucleus divide 3 times, 1 into 2, 2 into 4, 4 into 8. So by division 8 nuclei are getting, and this 8 nuclei arrange themselves. 3 move towards the micropyle, 3 move towards chalasa, 2 remains at the center. Cell wall formation takes place around each nucleus except at the center. The 3 cells formed at the micropylar side is called egg apparatus, with the central egg cell and 2 synergids. 3 other cells at the cells at the chalasa end are called as antipodal cells, and 2 remains at the center are called polar nuclei. This is an important diagram carrying 2, 2.5 or 3 marks. Diagram of female gametophyte. The matured embryo sac is called female gametophyte because it contains female gamete. So development of male gamete is called as microsporogenesis, that is present inside the pollen grain, and development of female gamete is called megasporogenesis. So the first part of fertilization called as gametogenesis, formation of male gamete and female gamete is over. Microsporogenesis and megasporogenesis is over. You have seen that pollen grains contain male gamete and embryo sac contain female gamete. You can take down the diagram. The diagrams are given in the notebook also. If possible, you can take down diagram from the video clips. See that. So first part is the pre-fertilization stage. The first part is the gametogenesis, formation of male and female gamete. Then comes the second part called as gamete transfer, or in plants gamete transfer is called pollination. Gamete transfer is called pollination. See, male gametes are present inside the androecium and female gametes are present inside the gynoecium. This contains female gamete, this contains male gamete, and this male gametes and female gametes should be, they have to reach together for the next process called fertilization. Male gametes are present here, female gametes are present here, and this male gamete should be transferred to the stigma for effecting fertilization. So for that, the male gamete should be transferred from anther to stigma. The transfer of pollen grain from anther to stigma is called pollination. Transfer of pollen grain from anther to stigma is called pollination. We have seen that in lower organisms, the gametes are motile, gametes have cilia. So gametes can move, female gametes and male gametes can move, and they can join and effect fertilization. And see, in plants, gametes are usually non-motile. They require external agencies are required for the movement of pollen grain from anther to stigma. Then we can see, before that, there are three types of pollination. See, there are three types of pollination. There are three types of pollination. First of all, we classify the pollination into three categories. First comes autogamy, second comes ditynogamy, and third comes allogamy. Autogamy, ditynogamy, allogamy, or otherwise called xenogamy. See, there are three types of pollination because different plants are there and different types of pollinations are there. First one, autogamy, ditynogamy, and allogamy. There are three types of pollination. In autogamy, I know the diagram, pollen grains are transferred, flowers are bisexually. Flowers are bisexually. So, pollen grains are transferred from anther to stigma of same flower. See, in autogamy, it is otherwise called self-pollination. Autogamy is otherwise called self-pollination. In autogamy, pollen grains are transferred from anther to stigma of same flower. So flowers are bisexual. You know what is the meaning of bisexual flower? Flower containing both androecium and gynoecium are called a bisexual flower. In bisexual flower, pollen grains are transferred from anther to stigma of same flower. Such pollination is called autogamy or self-pollination. And autogamy is of two types. Autogamy is of two types. One called cleistogamy and chasmogamy. Cleistogamy and chasmogamy. Cleistogamy means pollination in closed flower is called cleistogamy. In some of the flowers like plants like oxalis, flowers are always closed, they will not open. So in such flowers, pollination takes place. So pollination taking place in closed flower is called cleistogamy. Cleistogamous flowers. Because flowers will not open a cup. So such flowers, pollination of closed flower is called cleistogamy. And pollination in open flowers. Normally flowers are opening flowers in which anther and stigma are projecting out. Such for pollination in open flower is called chasmogamy. So there are two types of autogamy, cleistogamy and chasmogamy. Pollination in closed flower is called cleistogamy. Pollination in open flower in which anthers and stigma are projecting out are called chasmogamy. In cleistogamous flower, there is no chance of external pollen grains to come in. So in such flowers, in cleistogamous flowers, There is no other type of pollination, only self-pollination is possible because same pollen grains are falling on the same stigma, because flowers are always closed, so there is no chance of external pollen grains to come into the flower. So in cleistogamous flower, closed flower, self-pollination is an adaptation. The questions can be asked like that. In cleistogamous flowers, self-pollination is an adaptation. There is no other method of pollination. Cross-pollination will not occur. So self-pollination is only chance because flowers are always closed. So and there is no chance of external pollen grains. So there are autogamy, cleistogamy and plasmogamy. Then second one, geitonogamy. It is the pollination between neighboring flowers are called cleistogamy. See, I will just represent by diagram. Suppose flower, a plant. This is the transfer of pollen grain from one flower to the other flower of the same plant. See, transfer of pollen grain from one flower to the other flower of the same plant. See, pollination between neighboring flowers. One plant is enough. In one plant, male flowers are here, female flowers are there. Pollen grains are transferred from one flower to another flower of the same plant. This is also a type of self-pollination because one plant is enough for effective for pollination and fertilization. Pollination between neighboring flowers is called geitonogamy. Then comes the third one, xenogamy or allogamy. This can be called as cross-pollination. And cross-pollination or xenogamy means it is the transfer of or pollination occurring between two flowers of the same species. See, pollination occurring between two flowers of the same species. That is a must. Transfer of pollen grain from one plant to the other plant of the same species. You remember that? One plant to the other plant of the same species. So, pollination occurring between two plants of the same species is called cross-pollination or xenogamy or allogamy. So, there are three types of pollination: autogamy, geitonogamy and allogamy or xenogamy. And this autogamy is a type of self-pollination in which pollen grains are transferred from anther to stigma of same flower. And it is a type of self-pollination, and these are two types: autogamy, cleistogamy and plasmogamy. Geitonogamy is also a type of self-pollination in which pollen grains are transferred from anther to stigma of anther of one flower to the stigma of another flower of the same plant. One plant is enough in both cases of self-pollination. In cross-pollination, pollen grains are transferred from one plant to the another plant of the same species. It is called as cross-pollination or xenogamy. I think it's very clear. Autogamy, geitonogamy and xenogamy, allogamy. Autogamy and geitonogamy are self-pollination and xenogamy is cross-pollination. It is very important in the cross-plant breeding program because you are doing cross-pollination, crossing between two plants. In plant breeding, cross-pollination is very important because it will combine the characters of two plants together, two varieties together. We can cross the plants with different characters. So this is the basis for plant breeding or agriculture. See, so that's the types of pollination. And then comes the agents of pollination. So pollination and pollinating agents. Okay, what are the agencies of pollination? You know that pollen grains cannot move in or gametes cannot move in aquatic organisms, algae or some of the fungus. Gametes are in cilia. They can move. Male gametes and female gametes can come together and they can join and affect fertilization. But in most of the angiosperms, higher plants, gametes are non-motile, so they require the external agencies are required for pollination. We have studied that in some of the agencies birds are bringing out pollination. Pollination takes place by wind also. Different other birds or insects are bringing out pollination. When they are coming to collect the nectar or honey in the morning time, they will bring about pollination. So different agencies are there. Wind is there, water is there, insects are there, birds are there. Totally, the agencies of pollination, pollinating agents can be divided into two categories. So the agents which are bringing about pollination can be categorized into two and called as abiotic agents and biotic agents. We classify the agents into biotic agents and abiotic agents. The agents without life are called abiotic agents. Agents with life is called abiotic agents. And abiotic agents are two types. One is called wind and second is called water. Wind and water are the main abiotic agents. Insect, second one bird, and third one bat. They are the three biotic agents of pollination. So classified into abiotic agents and biotic agents. Abiotic agents include wind and water. Biotic agents include insect, bird and bat. And pollination by wind is called anemophily. See, pollination by wind is called anemophily. And pollination by water is called hydrophily. And pollination by insect is called entomophily. Study of insect is called entomology. Study of insect and pollination by insect is called entomophily. Pollination by bat is called ornithophily. Ornithology, ornithophily. And pollination by bat is called chiropterophily. Chiropterophily. So there are different agencies are there, but only something is some examples are given in the textbook. Anyhow, pollination is brought about by different agencies. They are classified into two categories: biotic agents and abiotic agents. Abiotic agents are mainly wind and water. Pollination by wind is called anemophily. Pollination by water is called hydrophily. And biotic agents include insect, bird and bat. Pollination by insect is called entomophily. Pollination by bird is called ornithophily. Pollination by bat is called chiropterophily. So these are the different agencies of pollination you have to study. And the terms are there, something related to that. And we have to see how the pollination is, what are the adaptations of this pollination and different agents. Flowers are having different characters are there, and we can see next class. How the wind pollinated flowers are certain characters, insect pollinated flowers are certain characters. That we can see the adaptations of pollination or flowers. We can see the next class. So I will conclude once again. We have seen the development of female gamete, how the female gamete or megasporogenesis takes place in the embryo sac. I said embryo sac is a 7-celled 8-nucleated structure, how the development takes place. Then comes the pollination, different types of pollination, autogamy, heterogamy, allogamy, and comes the agents of pollination, biotic and abiotic agents. The terms of the agents like that: anemophily, hydrophily, entomophily, ornithophily, and chiropterophily. Then see the adaptations are important. Adaptations of some of the flowers we can discuss in the next class. Then after we can see the second part of fertilization, reproduction called as fertilization. So I can continue the next class. So first of all, entire notes you can one class to diagrams you can in the notebook diagrams are given, and if you are possible you can take the diagram from. And after completing the portion, the 3 or 4 periods, I would like to conduct a test paper by any method. So write the notes on the same before listening the class, and listen the lessons and videos on the day itself possible, and ask doubts directly to me. Thank you.